Good afternoon, sir.
I welcome you. On behalf of CNRO Hall of Science and Education Technology Unit, once again I welcome you all for today's program, Learning Chemistry Through Experiment. We are having 45 minutes session followed by question and answer session. During the session, I request you to mute your camera and microphone. We urge you to actively participate in question and answer session. And you can ask the question directly, or you can comment your questions in the comment box. At the end of the lecture, I will provide the one feedback link, and you just use it. For YouTube participants, please mention your name and college name in the chat box. And thank you for your cooperation. So let me begin with the by introducing our speaker, Dr. K. Pramoda. Is a research associate and POSE coordinator at Jawaharlal Nehru Center for Advanced Scientific Research, Bengaluru. He obtained his Master of Science from the Mysore University and completed Doctor of Philosophy from Jawaharlal Nehru Center for Advanced Scientific Research under the supervision of Bharat Ratna Professor C.N.R. Rao. He worked as a visiting scientist at Melbourne Institute of Technology, Australia. He published more than 30 research papers in international journals, and he currently doing research on 2D nanomaterials and their composites for water splitting reactions. With this, I request Dr. K. Pramoda take over the session. Over to you, Pramoda.
I welcome you all. Thank you, Vinay, for the kind introduction. Firstly, I would like to thank Bharata Ratna Professor C.N.R. Rao and Mrs. Indumati Rao for giving me this opportunity for the live demonstration. And here at our center, JNCASR, we conduct number of programs for school and college children in physics as well as chemistry. I urge all of you to join in the future sessions as well. We can start PowerPoint. Firstly, I would like to start this demonstration with what we call famous words of Professor C.N.R. Rao. What he said is like, if you need a far excellence in science, what we need is a very good, strong, basic background. This we can achieve only, I hope this live demonstration will help in understanding the basic of things, and we can see redox oxidation, and so when we proceed, you will come to know, like, I hope this will help you in this. And Professor Mr. Vinayak told me we have a few good number of 10th students. So along with the first PUC and second PUC students, what we do, we'll go with little basics of chemistry, like acids, bases and salts. Then we'll move on to other experiments. So that what we're trying to do here, we'll just go for the basics first. We all have heard of acids, bases and salts. Here we can see I have named a few of them what we use in our lab, and whatever we use in our daily lives also, we can, depending on their pH, we can classify them as acids or bases. What we do, for determining whether it's an acid or a base, we have two important tests. One is litmus paper test that we are going to demonstrate now, and another one is phenolphthalein indicator test. First we'll go one by one with the experiment, then we'll start. So can we focus here? First what we have is like hydrochloric acid. You know, it's a mineral acid, and what we do here is like you can see it's a litmus paper. Okay, it's focusing. Okay, so if we dip in acid, color you can see changes to red color. Same paper, if I dip in the bases, so that it will give blue color. You can see the difference. So in the base it will give blue, and in the acid it will give red color. So according to this, we can say whether the solution pH is basic or whether it's acidic, or if you put it in the distilled water, it will be neutral. There will be no color change. That's what that's what we can see. See, there will be no change in color. So by these tests, whether we can see whether it's acidic, basic or neutral. So another important test is like phenolphthalein indicator test. Can we go for the slide? So here you can see This is a very important test for the acids and bases. It's basically a acid-base indicator. What since is programmed for the school children, I don't want you to straight away know all these structures. What we should know is like it changes color in oxidized and reduced form, and it shows color change according to the pH. So we already had a test of litmus paper. What we do, it's an acid, you can see, and we have a phenolphthalein indicator here. So we are adding few drops of phenolphthalein indicator to the acid. You can see, you can't see any color change in this. As soon as in the PPTs you can see, there is in the acid medium there is no color change. It will be colorless. As soon as if we start adding base, you can see pink color appear. So another one important point is like how salts are formed. Okay, so salt can be like when acid combined with the base, it gives salt and water. That's what we are doing. What is it? What we have a little acid and phenolphthalein indicator just to show how it changes the color. See, you can see pH. How can I say this is acid? I already told you. If I put a pH paper, it should be there. Okay. So, and we have phenolphthalein indicator. As soon as I add base, you can see pink color is appearing. See, so when we get a color, it means indicator is in the this form. You can see. So that pink color is due to the another form of the indicator. It's the its pH is more. You can see. You can test the pH now. The solution might not blue color. Is it appear? So these are the two important tests what we do at our laboratory for the acids and bases. And I already told you there are number of acids what we use in lab and what we use in our daily products as well as what we consume also. So next one we are moving on to here basically we are not explaining much like we are demonstrating through the live experiments. And next one I would like to go for Lee-Chatelier principle. So we all heard of chemical equilibria. So what is chemical equilibria? You can see from this reaction. See, it's a reaction where chromate will go to dichromate. So if you see, this chromate ion is in the yellow color. If you put acid, it will become dichromate. So I'm not going to much structure of this. So this basically this color changes you can see. This almost this chromium oxygen bond will be double bond. This will be chromium and oxygen bond will be single. That's why we see change in color. First what I take here is so this is the solution where we dissolved chromate in the distilled water. So see, this is the reaction where we have chromate ions in the distilled water. can see I will put a few drops of acid. Basically it is a nitric acid. So you should be careful with the nitric acid. You can see the color change now. So when we put acid, what's happening? It's like enough. It's like you see, reaction is shifted to right side. Again, what we are going to do, we are trying to add a base. What base does? What we know in the earlier reaction, it neutralizes acid. So if it neutralizes acid, it should come back to yellow color. So we are adding a few potassium hydroxide pellets. It takes some time to dissolve. You can see the color change. See? So chemical equilibrium reactions, we can give a number of examples. We are taken here chromate to dichromate ion conversion. What we observe, see, when we add acid, it will go to the dichromate ion, which will be in the red color. So again we added base, it will come back to the yellow color. It means it's in the chromate ion. Suppose again if I add acid, it will few drops of acid. If I add, because we added more base, so we need to add more acid. You can see the color change now, right? So when solution is acidic, so it go back to dichromate, and when solution is basic, it means if it neutralizes acid, it will be in the chromate form. We can give examples like many examples of chemical equilibrium reactions when nitrogen and hydrogen combines to give ammonia. That's also kind of chemical equilibrium reaction. You can, like, by changing the acid or basic conditions, we can play between forward and backward reaction. Next, I would like to go to more interesting experiment. That's a blue bottle experiment. So this I already told you about indicators. You no need to much worry about structures, whatever we use. Just what we go when we study this, these materials will be recorded in our mind, and you just need to know when it's oxidized, it will be in the different form, and when it's reduced, it's in the other form. How we know in the solution, they give different colors. You can see here. So first, what I do here, it's a blue bottle experiment. So basically, we are see, it's a distilled water, and we are adding 5 grams of sugar to it. Sugar means glucose. I am adding 5 grams. So we are dissolving glucose, and you can see what we are doing here. So glucose we are dissolving in distilled water, and we are adding to it potassium hydroxide. So glucose contains aldehyde group, and we know in presence of oxygen it will convert to acid. So, and also for this we are adding KOH. KOH, potassium hydroxide, is kind of a catalyst. It just catalyzes the aldehyde conversion of aldehyde conversion to acid. You can see it needs to dissolve totally. So what's happening is glucose is converting into gluconic acid. So what the water we are using is distilled water here. Then I will come to indicator part. Here you can see it's a methylene blue. So we are adding 2-3 drops of methylene blue. You can see initially dye is in the, is it blue color is visible, dye? You can see it is blue color. You can see strong blue color. And I will keep for some time. You can see what happens in the reaction. Is it focusing here? You can see. So after few minutes, this indicator, whatever the color, it comes to colorless form. So this reaction, it will convert to colorless form. So what's happening in the reaction? So if there is a color change in the reaction, there should be something is happening. So what I told, when we dissolve this glucose in the distilled water in presence of KOH, it will convert to gluconic acid because of the oxygen. What happens when we add methylene blue? Initially it is in blue color. We know this it's in this structure. It's in the oxidized form. Why it's in the oxidized form? Because methylene blue is in contact with oxygen, so it will be in the blue color. As soon as we add it, what happens? This reaction takes oxygen from this environment, and because of this reaction, this indicator will get reduced. Again, see, when I shake it again, you need to. Is it blue color is appearing? When I shake it again, you can see. Again blue color is appeared. So you can think now what's happening. See, again when methylene blue in the reduced form, means it's in the colorless form. As soon as we shake the bottle, it takes this solution takes oxygen from this, and it will become blue color because of the oxidized form. So what happens when after shaking? If you see, once we leave the thing after shaking, it will become colorless again. So whatever the oxygen, it is used by this reaction, and glucose will keep on converting to gluconic acid. Again, this can be done n number of times. As soon as this dye comes in contact with oxygen, this blue color keep on appearing in the reaction. It says we have different oxidized and reduced forms of the dye, and that's what it gives. And we all know oxidized means it gave electrons, reduced means it receives electrons. So we can n number of times we can repeat this experiment. It's a very famous experiment. I think blue to colorless form is appearing. Any n number of times we can do. So I told already, once we shake this, so this methylene blue in reduced form means it's in the colored form. It will convert to oxidized blue color. So next we'll proceed to chemical trophic light experiment. It's quite similar to blue bottle experiment, but what this indicator here, I already told you, don't much worry about the structure of indicator. What we are trying to say, in the oxidation and in reduced form, these indicators will exist in different colors. Why? You can see the change. You can see the double bonds here. So there will be change in the conjugation, so that that's why they appear in the different colors. So for this also, what will take sugar. It's the same reaction. We dissolve glucose in the distilled water. So I already showed you in the last experiment. Glucose we dissolved and KOH is also added in this. So this is a reaction. So this is a indigo carmine dye. See, dye is in the blue color. You can see the blue color. And now I am adding think as soon as I add it, it turns into green color. You can see the green color. So see, as soon as oxygen is keep on reducing in the solution, so that it changes from green to red to yellow. That changes we have focus here. So same like the earlier experiment, this in the oxidation form, when we add it, it gives green color. And one more thing, you can see it comes to almost red now. It's visible. It's a mixed kind of. And finally, it will come to yellow. Why it will be in the yellow color? You can see this reaction it gives the electron, so it always try to be in the reduced form. Red is appearing now. This color change depends on the concentration of whatever the dye we add, and concentration of base as well in the solution. And one more change we saw, when we added dye, it's in the blue color, and as soon as we start, it becomes green. So can you guess why? Because dye it's in the neutral pH, because we dissolve that in the water. So when we add to this, it will be in the basic condition. So under basic condition, you can see it will exist in this form. So finally it comes to yellow color. You can see the change. As soon as it start losing the oxygen, it goes from green to red to yellow. So again, what we are doing, we are just here we have enough oxygen, and we'll just shake the bottle. So we are dissolving, we are giving enough oxygen for the dye, so that it should exist in the oxidized form. I think it's clearly visible. So as soon as this reaction keeps taking oxygen from this, so the dye will be keep giving oxygen, and it will be exist in the reduced form. That's what Okay, now it will be in the green color. It convert to red. It convert to yellow. And one more question I want you to ask people: how long this reaction can go? So there are two things here. One is like till all the glucose will convert to gluconic acid. This color change we can observe. And another thing is these dyes are like they can decompose under photochemical conditions. So till the dye is intact, we can get these color changes in the reaction. I think it comes to it is in this reduced form, means red color. It's kind of a intermediate. Then it will come to yellow reduced form. So this basically we try to explain how color changes. We can know we can tell the how whether reaction is happening or not happening. There and in case of phenolphthalein, it's like it tells us how pH changes in the reaction. So next we are going to we all heard about Michael Faraday. So he conducted he gave us laws of electrolysis. So and nowadays quite number of research is going on water splitting. It's like generating hydrogen from by splitting water. So we are going to demonstrate how this electrolytic cells work during water splitting reaction. So we have a setup here. Uh, we just do with a two. You can see pencils contain graphite. Those we are used as a why graphites are electrode, because they are electrically connecting. They can as electrode. What we add some Epsom salt, because you know we are the cells here we are using distilled water, not conductive. So we need to that's why we will add Epsom salt. Epsom is magnesium sulfate. We are just adding distilled water. So water is this distilled water is non-conductive. To make it conductive, we'll add Epsom salt. Is it focused? Is it focused? So we add little Epsom salt to make it conductive. So the Epsom salt, what we added is kind of a neutral salt here. It does not involve in any of the reaction. So for this, we want to know, need to study petrochemical reduction potential of various ions are given. So what I am trying to say here is like Epsom salt is, does not involve in any of the electrode reaction. I told you we have used pencils, let's say as a graphite rods. Just through simple experiment, we want to explain how this water splitting can take place. It's very important reaction nowadays. Hydrogen can be used as a clean fuel in the fuel cells for running the motors. So what we are doing, we are connecting this graphite electrodes to a battery for the power supply. You can see this is the circuit. So, and positive terminal of the battery is anode and negative terminal is cathode. So as soon as we start connecting, please focus here. You can see bubbles, you can see. I think you should focus. Bubbles are started coming. See, the thing is, there is something to do with uh, one side it's bubbling fast, right? Another is very slow. See, you can see this chemical reaction. For every 1 mole of water, one mole of hydrogen will be released and half mole of oxygen will be released at the uh, anode. So whatever fast we are getting, fast bubbles, is from the hydrogen, which is cathode, and oxygen, which is the anode. And Epsom salt is kind of a neutral salt here. I think as soon as we'll connect power supply, it's keep going. It's a very simple experiment that we can conduct at our homes as well. So bubbles, as soon as power supply is there, bubbles keep coming. So next we will move on to electrolysis of sodium chloride. These bubbles are. So in the before we start, we make the setup. In the earlier we shown this whatever we added Epsom salt is neutral under this condition. Just to know whether salt will react or it is neutral under this condition, we need to look out the petrochemical series that I am not going to discuss here. So what we do now is like instead of Epsom salt, we dissolve sodium chloride in the solution, and we'll see all other things we keep same. Let's see what happens in the reaction. And we are setting it up. So we separately took two beakers in this case because we wanted to show this whatever hydroxide whatever we dissolve sodium chloride, it will produce sodium hydroxide at the cathode. For this, if you go back to our old slide, you can see how this in the basic conditions, this our dye should give pink color. So will show that experiments like whether sodium hydroxide is produced at this cathode or not. So we have a sodium chloride in both the beakers. Instead of pencils, we use you can use either pencil lead or the graphite rods directly. Both are sodium chloride. Both we are used graphite rods as electrodes, and will potassium nitrate. So we all heard about this salt bridge. just if there is any to maintain electrical neutrality, what we do, we just simply made through the cotton this salt bridge. Just what we do, we prepare a potassium nitrate solution. We can show here. We prepared potassium nitrate solution, and we dip this cotton just to maintain the electrical neutrality. That's what we read. If there is any change in the ion concentration, it just neutralizes this. Just we connect that cotton to the both of the beakers. Make sure our salt bridge is not touching to the electrodes. It should be separate from that. And we'll connect it to a power source like earlier experiments. And I'm adding phenolphthalein indicator so that if it is in the basic form, it pink color should come. Otherwise, it is no color means there is nothing basic conditions. So we'll see whether this reaction happens or not. Is whatever we have PowerPoint. Whether this sodium hydroxide will get released, and we can see hydrogen at the cathode and chlorine at the anode. Can you focus here? You can see as soon as we started connecting, this pink color is appearing. Just initially it is in colorless, so pink color started appearing. It suggests solution became basic. So we already shown in the phenolphthalein indicator. Yeah, if we give more time, solution will become more pink. So we need to give 2-3 minutes. Phenolphthalein indicator I am just adding. You can see pink color is appearing. That means sodium hydroxide is producing in the solution by electrolysis. So I think what I suggest you, since as we were school children, we have gone through some of the structures. You no need to remember all these structures. Only thing is like you have to remember it exists in different colors. So as soon as the color changes, that that suggests reaction is happening and oxidation, and it's in the oxidized and reduced form. And simply whatever we use at our homes, like whether it's in alkaline, acidic, or basic condition, we can just know by litmus test or pH indicator test. So with this, I would like to thank Bharataratna Professor CNR Rao and Mrs. Indumati Rao for their constant support. And we have number of programs. I have just given. We conduct number of programs in at JNC as well as outside, and their vision and commitment towards popularizing science is unparalleled. So thank you very much, sir. Uh, and I would like to thank Praveen for helping me with the experiment. Professor T. Govindraju, who is always a constant source of inspiration. And I thank Mr. Vinayak for this wonderful opportunity. Thank you, Vinayak.
Thank you, everyone. And thank you, Pramod sir. With this, I thank you once again, Pramod sir and Praveen for helping him. And it was a very interesting demonstration. And now it's time for a question and answer session. If you have questions,
Should I come?
yeah, just go there. Any questions? For online? Okay. If you have questions, you can send an email. And right now, if no more questions, let's thank Dr. Pramod sir. And it's end of the session, and it's with the vote of thanks. On behalf of CNRO Hall of Science and Education Technology Unit, we express our sincere thanks to Dr. K. Pramoda for giving us such interesting and doing the demonstration. I thank Praveen for helping him for experiment. We thank the President of JNCASR, Professor G.U. Kulkarni, for his extending his support and facilities for this particular program. We thank the Mr. Dwarkanath and Narayan Rao for coordinating with the schools and colleges for participating in today's program. We thank the principals, students and teachers from the various colleges who are participating in today's program. Especially principals of JNV Yadgiri, JNV Ramnagara, and JNV South Kendra, Mangalore, and Maharani Lakshmi Ammani College principals and all. And thank you once again. And we thank the supporting staff, administration of JNCSR providing the infrastructure, facilities, and administrative support for this program. Thank you. Thank you, one and all. And last one thing I wanted to mention, and I have given the feedback in the group in WhatsApp group, and please fill the feedback, your valuable feedback. And here in the YouTube live, and there is a description. You just drop down arrow is there in the description. You can see the feedback link, and it will open at 3 o'clock. And just give the feedback. And thank you one and all.
ಇನ್ನು ಸ್ವಲ್ಪ ಸ್ಲೋ ಬರಬೇಕಿತ್ತ ಓಕೆನಾ? ಕಲರ್ ಎಲ್ಲ ನೀಟಾಗಿ ಕಾಣಿಸುತ್ತಲ್ಲ ಮುನೇಗೌಡ್ರೆ?